You're all so quiet. Wow. Okay. So I wanted to continue on with plant cells and tissue, and then get into plant growth. That's next slide that posted. And hopefully today, when you go into lab, you'll be able to see that your seeds have germinated, and you'll have radical and organic roots coming out. But if you don't see that, check to see how many overall. It looks like it's increased in size. Where you start probably safe to plant. So, okay. So yeah, fingers crossed on that. All right, so looking at complex tissue in terms of the vascular tissue. So the artist here drew the vascular tissue in blue and red. So we have to think about the vascular tissue. It is analogous to the blood vessel system of humans. But there's going to obviously be some key differences in terms of how it functions. But right now, I just want to introduce kind of the anatomy of the vascular tissue. And the first thing I want to talk about is the xylem. And so xylem is very important because that's how the plant gets access to water, in the soil, and also any minerals that might be dissolved in that water. And so the roots have a very, very important job besides stabilizing the plant, keeping it rooted in place. Also, all those roots have to take up water. And then it dumps into the xylem. And the xylem courses through the entire plant body all the way to the leaves, the veins in the leaf. So then we're also going to look at phloem. So xylem and phloem, when you look at it, you look at a stem, let's say, in cross section, you will see xylem and phloem right near each other. They tend to be clustered near each other, and you'll start seeing that quite a bit in lecture and lab, and get used to that when we look at a lot of different slides showing different parts of the plant body. So let's get into, we'll get into the phloem as well, but let me focus on the xylem first. So xylem, when you look at angiosperm flowers, they have vessels. So this is a subtype of xylem. So there's different kinds of xylem. So one is the vessel, and they're open at the ends, and they stack end to end to end, form these long systems of xylem cells that start in the roots, go all the way to the vein. Imagine stacked end to end. They're open. They're dead at maturity, so that means they have secondary cell walls. Okay, so they're very, very rigid and thick. And the vessels in particular tend to have a pretty nice wide diameter. That's important. So vessels are wide, stacked end to end. They're dead. There's no cytoplasm in there. So it's a very efficient way for water to reach the plant body. There's no cytoplasm flowing down the flow of the water. So the images that you saw, go back to that. You can see that the vessels, there's a nice one right in the middle, a really nice example of a wide one. And then you can see that they have, like, sometimes they have these perforations at the end of the cells, but they just stack end to end to end, okay? With the other type of xylem I want you to be familiar with are the tracheae. Tracheae. Angiosperms, the flowering plants, also have tracheae, right? They're thinner in narrower, smaller in diameter compared to vessels. And so when we look at different parts of the plant body, I'll point out which ones are vessels, which ones are tracheae. They look visually different due to their differences in diameter. They tend to be more tapered at the ends as well. But again, it's the same basic idea. They stack end to end to end. And then you have to imagine that going all the way from the roots up into the veins of the leaf. And so it is a continuous system of all of these xylem cells, whether they be vessels or tracheids. Dead at maturity again, so that means they have a secondary cell wall that's pretty thick. And then the other part of the vascular tissue, when you think about like big trees that have really wide tree trunks, they'll have rays that will radiate out from kind of the center of the tree trunk out to the periphery. And so that's just for lateral conduction. And so that lower image is, you can see that kind of striping pattern of the rays. So it's just a kind of an avenue or a route for lateral conduction of water and can help with food storage. So those are made of parenchyma cells. So the main part of the xylem tissue that I want you to focus on are the tracheids and the vessels. I don't worry so much about the rays, but I know that many of you have seen a cross-section of a tree trunk before, and if you've ever noticed those kind of radiating bands, that's what those are called, the rays. Okay, so xylem is tracheids and vessels. Angiosperms have both kinds, right? With the gymnosperms, the conifers, long gymnosperms, they really only have tracheids. Right, so we consider the gymnosperms to be a little bit more ancient, more primitive. They really only have tracheids. There's just a couple of exceptions to that. The angiosperms are considered more advanced, more evolutionarily advanced, and they have both tracheids and vessels. And they think that's one reason why the angiosperms, the flowering plants, are so successful. One reason is that they evolved those vessels, which are wider in diameter, allow for more efficient water movement through their body. So we'll talk more about angiosperms and their successes in this class for sure. Okay, phloem tissue, what do we have there? So xylem and phloem tend to cluster together. Phloem tissue is also super, super important because this is where we see sugar being transported. So the sugar dissolves in water and then it moves through the plant body. They are actually living cells. They don't have a secondary cell wall. So when you look at them under the microscope, their cell wall looks much more delicate, much thinner compared to the cell. They're modified, like they don't have a nucleus, which is weird. I mean, you've been taught since you took intro biology, all cells have a nucleus. Well, here's an exception, right? So fib tube members are what we see in the angiosperm. So they can be called sieve tube members. Other people will refer to them as sieve tube elements. Sieve tube, at least that's basic. But sieve tube members, sieve tube elements. They're living at maturity, so they don't have a secondary cell wall, just a primary cell wall. And then this drawing is trying to show where they stack end to end, where two cells meet. There's this region that they call the sieve plate. And they have it pointed out here in this part of the picture, okay, in the top part, the sieve plate. So those, it's like a sieve, like, you know, there's a bunch of pores in there. It's all open, there's nothing blocking that. They're just hollow pores at the end of the sieve tube members. And it just allows for good movement of the sugar that's dissolved in the water from cell to cell to cell. So sieve tube members are what we see in angiosperms. So angiosperms being the flowering plants, they're the more advanced plants, evolutionarily speaking. All right, so we have those angiosperms. Now, something else on this drawing that I want you to key in on is the companion cells. So these kind of yellowish, I guess they're yellowish, that are right next to the sieve tube members. So these companion cells are super important because I told you that the sieve tube members don't have a nucleus. So they're not fully functioning. Sieve tube members aren't like capable of surviving on their own. because they lack a nucleus, they lack some other organelles that I'm not going to get into. So they're highly modified. And if you try to keep them alive without the help of a companion cell, it doesn't work. And so these companion cells, they named them appropriately, they're a companion to the Civ2 member. They help the Civ2 member with their physiological needs. So the companion cell is busy doing what it needs to stay alive, plus it does give a lot of the resources that it has to its neighboring Civ2 member to keep it alive. And so it can be sometimes challenging to see under the microscope, but it's a one-to-one relationship. For every Civ2 member, there's a companion cell right next to it. And they've experimentally gone in with a needle and they'll kill a companion cell. And then within minutes, they see the Civ2 member that's right next to it dies as well. So the companion cell, they think, is absolutely necessary. There's lots of plasmodesmata connections that will allow for flow. Remember the plasmodesmata are those internal tunnels between cells, between neighboring cells. So you see these plasmodesmata, lots of them, that are going between the companion cell and the Civ2 member. Now, in the gymnosperms, we don't see Civ2 members. We see Civ cells. They're different. They function a little differently. They look a little different. And we see them in the more primitive vascular. So gymnosperms would fit that category. And then you're going to learn about seedless vascular plants like ferns. Ferns are a good example. Seedless vascular. So really, there hasn't been a ton of research done on Civ cells. So there's a lot of information that still needs to be discovered about them. They're, like I said, they're not even quite sure exactly how they function. And so they're still needing to work on that. By far, most of the research done on plant physiology is with the angiosperms. They get a lot of the attention. All right, and then let's zoom out to the very surface of the plant body. So just like we have an epidermis that covers our body, we also see that with plants as well. Usually 1 cell thick, and often, depending on the plant, you'll see variation, but almost every plant that I can think of, can't think of an exception, will have some kind of waxy surface covering. And they often refer to it as a cuticle, especially when you think about leaves. They'll talk about that waxy covering and they'll call it the cuticle. Right? That's just the name that they give it. So here on this image, you're seeing that pinkish layer. That's all the waxy, it's very hydrophobic. So basically this waxy substance minimize any water loss from the plant. It's like waterproofing the plant. And if you think about it, that's a pretty good strategy because a plant can't get up and go to the refrigerator for more water. All right, it has to be very, very careful about its water supply. And so covering most of your body in wax and then restricting water loss from your body to very specific points, that's a good strategy. So that cuticle can be thick or thin depending on where the plant is living and what kind of habitat it's exposed to. We'll talk more about that when we get into leaf anatomy in more detail. Here you're seeing a drawing of a leaf, and you can see that there's an upper epidermis, 1 layer thick with that cuticle covering it, and then there's a lower epidermis. All right. And then you can see on the lower epidermis, they have these green cells scattered. So those are the guard cells. And the guard cells guard the pore in between them. Typically, they're kidney shaped, like what you see here. They look like kidney bean seeds, shaped like that. And then that pore is in the middle. So taken together, the pore and the 2 guard cells, we call that a stomata, or stoma for singular, stomata plural. So stoma is, if you just add a TA to the end of that, that would make it plural. And the way the artist has drawn this, you can see that the guard cells and the pores, they're only on the bottom of the leaf. They're not on the top of the leaf. And that is a strategy that many plants employ. They'll tuck their stomata on the underside of their leaves. So the way the artist drew this, I knew that they were thinking probably about a plant who has its leaves mostly, for most of the day, horizontal And so if you think about it, that's a pretty good strategy, because as the sun comes up, I guess if that's north, sun comes up in the east. And then if you think about high noon and the sun beating down on the surface of the leaf, it makes sense that they tuck the stomata underneath, so that if you can imagine heat hitting the top of the leaf versus the cooler underside of the leaf, if you have a lot of stomata on the top of the leaf and the sun is beating down on that part of the leaf, you get much quicker loss of water out of the stomata. And like I just said, plants are pretty careful about their conservation of water. So it makes sense if they put the stomata on the underside. It's more humid, it's cooler, and that will slow down the loss of water from their body. So it's a good strategy. We'll talk more about where plants put their stomata as we get more into leaf design. So here you're looking down on the epidermis through a microscope, and you can see the guard cells. Hopefully you saw that yesterday, I think, with maybe the aloe vera leaf. You took a thin section and we're looking at that. And I think we also had some prepared slides. And it's, I mean, the really cool design. So all those like jigsaw looking things, those are epidermal cells. It's like a jigsaw puzzle. And then you see the darker kidney bean shaped guard cells that are guarding the pores. So that little white slit is the pore in the middle. So the pores are open a little bit. And we'll talk more about how the guard cells function to regulate the size of that pore. So guard cells can make it so that the pore is like 100% open, or they can get it down to like 10%, 2%, 55%. They have lots of sophisticated control on how wide should that pore be. And it's always being in response to what's going on in their environment. How much water do they have access to? And that information will feed up to the stomata and tell the guard cells what to do. Another part of the epidermal layer is the idea of the hairs that you see, trichomes. So you'll see that today in lab. We'll have some different leaves for you to look at under the dissecting scope. And trichomes, I like you to know that term. Most people just say, oh, look at that hairy leaf, and they see all the fuzz on the leaf. Those are trichomes. So please know that term. And so they're just epidermal cells that have modified to come off the leaf in a long, skinny morphology. They're still epidermal cells. They're just modified. All right. And trichomes have a great diversity in how they look, how they feel. Some are soft, some are brittle and hard. Stinging nettle, if you've ever had the misfortune to bump into stinging nettle, that's almost like a hypodermic needle trichome, and it injects the toxin into you that makes you itch and swell. So not much fun, but there's other trichomes that are very soft. You're going to look at cotton, where we get cotton from under the microscope today, and those are trichomes. I don't know the history of that. I'll have to look into it. I just think, who thought of that? Let's take some cotton seeds, strip off this fluff, and make cotton fiber out of it and fabric. It's kind of bizarre, but it works. Cotton has been very, very successful in industry. So here you're seeing again the waxy covering, the pink, the cuticle. And then you can see here they have two guard cells surrounding the pore. So taken together, that whole thing is called a stoma, right? The guard cells in the pore. And so they often refer to that wax as cutin. Okay, they'll say, oh, look at that cutin wax making up the cuticle. That's how they would refer to it. All right, and again, that thickness can vary, but really, really good at waterproofing. And then really the only water loss, because of all that wax, really the only water loss is through the stomata. And the guard cells can control that quite precisely. So it's a nice strategy. There is also an extra benefit to that waxy covering, because it's a little bit more challenging for pathogens to make their way through that. It's a pretty complex polymer. And so fungi and bacteria, let's say, they have a little harder time getting through that if they want to try to attack the plant tissue. So it does provide that benefit as well. And then here you're looking at root hairs. So root hairs, again, we're still on the epidermis of the root now. And the epidermis you have epidermal cells of the root, and then some of those epidermal cells come off in that long skinny shape again, kind of like the trichomes, but now we're focusing on roots. And so then we call them root hairs. There's no fancy name for them. We just say root hairs, okay? So all of these things projecting off the surface, they're just epidermal cells. It's just that they were modified to come off. And then on the left, you can see a very, very young seedling that just germinated. And very often look for that today. Hopefully, if you have some roots coming out of your seeds, look for the root hairs. It's pretty impressive typically. And the root hairs really are great for the plant because that shape, that long skinny shape, increases the surface area really nicely. And so it's much easier now for the plant to take in the water from the soil. So it's really the root hairs that do the lion's share of the job of taking up water because of their design. They have that really large surface area, soak up the water. That's why if you have ever read directions on when you transplant a plant from, let's say you're going up a pot size, you your plant out through your pot, you want to put it into a slightly bigger pot. And usually in the directions they'll tell you, make sure to water your plant frequently right after you transplant. And that's because no matter how careful you are, typically you destroy a lot of these very delicate root hairs during that transplanting process. And so you've made it harder for the plant to take up water. They're not as efficient until they regrow those root hairs. So that's why they're telling you, make sure to water when you transplant. Okay, another layer, outer layer that I want to talk about, at least introduce, is the periderm tissue. And when we get into stem tissue, we'll talk more about this, but I want to introduce it now. So the periderm is what they refer to as the plant is maturing and you start seeing bark production. Then what's going on there is the epidermal cells are getting replaced by the periderm tissue. And it then will allow for bark formation. And I'll talk more about bark in detail, but basically like the outermost portion of the bark is called the cork cells. And say we have lots and lots of cork cells that make up that outer layer of the bark. And cork cells, once they are fully mature, then they die off. So they have a thick secondary cell wall. And then most of the time, they're really well layered with a different kind of wax that we call suberin, not cutin. Cutin tends to be more for leaves, and then suberin tends to be more for the stem tissue. Okay, so suberin, but again, same basic function. You want to waterproof that outer surface of the bark so that you don't have water loss. It can make it a little bit more challenging for pathogens to get in as well. So same basic idea. However, tree trunks still need ways to get access to oxygen in the air surrounding them because there's living cells in stem tissue that need oxygen, just like we need oxygen. And so the image here of the tree trunk, you see those like white dots along the tree trunk. Those are what we refer to as lenticels. Lenticel. And if you'll see this under the microscope when in lab, we get to this section in more detail. But basically lenticels are just discrete regions along the tree trunk of loosely packed cells. So it's just loosened up in that area, but it's discrete. And what that does is it's a little bit looser cell pack. They're not packed so tight together, so there's more air spaces in that area. It just allows for atmospheric oxygen to come in those regions to the tree trunk so that the oxygen can get to the cells and those cells will stay alive. So it allows for gas exchange, oxygen in, CO2 out. And really, it's a way for the stem to breathe. So we'll look at that in more detail when we get into stem tissue. But here, let me show you. This is what you would see under the microscope. So you're looking in cross-section of a stem. And you can see here this little bumped region that's very different than what you see on either side, where it's more packed together. Then all of a sudden you come to this region that's kind of loosely packed. So that whole thing is the lenticel. All right. So that allows for gas exchange to come in. Okay, so I've introduced a lot of terms, and it's just like in any discipline that you learn, there's going to be jargon. So there's been a lot of jargon for that, and so you might want to do flashcards and do that kind of system for, I mean, some of this is just basic memorization where you have to know what these terms mean. All right, so make sure that you're studying a little bit every day, focused studying, 20 minutes or so, take a break, and try to get these terms into your long-term memory. By the way, if you do flashcards, especially if it's a physical flashcard, I don't know if they can do this on the flashcard apps. What's that flashcard? Quizlet. Do they let you shuffle? Good, shuffle. Do that. Because if you keep seeing the things in the same order, your mind goes on auto. You're really not truly learning. You're just kind of half there. So shuffle the deck so that it's a little bit more challenging for you. Okay. If you have your laptops with you or some way to access the EduCat, I'd like you to answer a question right now. So if you could get, and if you don't have your laptop, you can but I want to ask you a question. And it's going to be in EduCat. So you can get into my EduCat page. At the differences between plant hormones and like animal hormones that we have, there's some differences, but there are definitely some similarities. So basically, when you look at hormones, I give a lot of kudos to the plant physiologists that study plant hormones. It would drive me crazy because they're produced in tiny quantities, and one hormone can affect many different developmental processes. And then you can have more than one hormone doing, all involved in one process. It can get very, very complicated. But in general, some rules of thumb to think about: hormones are produced throughout the whole plant body. It just depends on which hormone you're thinking about. But you can have one hormone being produced at multiple parts. So in general, when you look at animal hormones, there's usually a discrete region of the body where that's produced. With plants, it's much more diffuse. You can see many, many different spots of the plant can have the same hormone being produced. Hormones in the plant can act locally or at a distance. Typically, I think most animal hormones do like more at a distance action. Plants, though, they can make a hormone in one spot, it can have an effect there, but then they send the hormone somewhere and it can have an impact at the distance. One hormone can influence many processes. Absolutely true. In general, when you look at, okay, here's a hormone coursing through the plant body, it's reached its target tissue or cell, then what tends to happen is you have the hormone chemical actually docking into the receptor protein on that cell surface. And so that tends to be the routine, all right? So the chemical hormone will dock into the receptor protein. They often refer to this kind of interaction as the lock in, or the key into the lock. It's about a locked door. The key is the hormone, and the lock is the receptor protein. So hormone work actually, believe it or not, it actually all began, really, or at least was credited with the beginning of hormone research, is Charles Darwin. Charles Darwin and his son Francis were avid botanists. They were way into it. Like their home was a botany laboratory, and they did a lot of tinkering around. And then when they started doing more tinkering, they got more serious about it. And so this top yellow box is some of the research that initially Charles and his son Francis started working on. So here, basically, and this all started with, they noticed the plants that they had in their windowsill would bend toward the window if they just left them alone. The stem and the leaves would face the window. And that they just were like, huh, why is the plant doing that? How is the plant doing that? And so they started to do some research. So here they're working with what we call coleoptils. Those are just, I don't remember which plant this was, but a coleoptil is just a sheath of tissue that covers the seedling as it's emerging from the soil. And they're very young and flexible, so it's easy to get them to move under these conditions. So what they did was they had unilateral light shining on one side of the young seedling, just like coming through a window in your home. And when they did that, they would note that the seedling would bend toward the light. So then they're like, well, I wonder if there's some kind of signal coming from the tip of the plant. So they amputated the tip of the plant and did the same experiment. And you could see that there was no bending toward the light. So then they're like, oh, well, maybe there's some chemical in the tip that is necessary to get the bending to occur. So then they said, or, maybe us amputating the plant really messed with it. That's what's going on here. So then they said, okay, let's not amputate. Let's just cover the tip so no light can hit the tip. And when they did that, still no bending. So like, okay, seems like light is necessary to hit the tip. And then you get something going on that causes the bending. So if you do cut the tip off, would you get this kind of bending? Well, it slows down primary, like lengthening of that particular stem would basically stop, at least for a while. You'd get lateral growth, like the axillary buds would become active and you would get a bushier plant. You'd still get growth. It's just in a slightly different manner. So yeah. And then they said, well, is it just the tip that we need to think about? Okay, well, let's bury most of it in this loose soil, and let's do that again, where just the tip is getting hit with light. And when they did that, they noticed a bending. So like, okay, definitely the tip. If we hit the tip with unilateral light, it'll bend, the seedling bends toward the light. So, okay, so that's, and so that's pretty simple experimentation, no fancy technology back in the 1800s, 1880. So that was a long time ago. And so they were able to, get some data from that. And then all these other rows on this table, you can see other scientists came along and added their experiments into the pile of information. And I'm not going to go into the rest of them. You can certainly look these over. But basically, again, just adding to the idea that there's something going on in the tip here. And they ended up realizing that it's auxins that are there. Now, Charles and his son Francis did not name that hormone. They're not credited with that. But what they found is that auxins are produced in good amount in the apical meristem tissue of stem in particular, right? So stem tissue in particular. But what we see here is that that auxin hormone really stimulates cell growth. All right. So hang on just a second. All right, I'll get to that. So let me come back to this. Whoops. Let me go back. One more. All right. So the actively growing part of the plant. So the meristematic tissue in general, you generally just see auxins. But for sure in stem tips, you get a really good dose of auxins being produced there. So when you think about the actively growing areas, we're usually focusing in on the meristematic tissue that you can find in different regions of the plant body. So they have many effects. I can't go into everything, but they are kind of like a master hormone in some respect because they can control the production of other hormones. So they can interact with, okay, we need this other hormone up and running as well in order to get some development to occur. Right? So they can trigger the production of other hormones. They've done a lot of research and they now understand that auxins are necessary for proper fruit development. So of course, for a plant, fruit development is super important because that's where the seeds are located, where they, the plant has put its genes into the next generation, into the seed. And so this experiment that you're looking at here in this image, here's a strawberry, a typical strawberry. And actually, when you eat a strawberry, what you're really eating, all that red sweet flesh, it's actually modified, the base of the flower modified into the red sweet flesh. So you're eating mostly flower tissue when you eat a strawberry. And then all those seeds, we often say, look at the seeds on the strawberry. Technically, they're dry fruit. So we're going to learn more about that. But a strawberry, actually, every single seed is a dry fruit. And they're called achenes. The fancy word is down there, achenes. So you're eating mostly flower tissue with a bunch of dried fruit on it. It's kind of weird to think about it that way, but fun to think about it. So what they found is if they do an experiment where if you get a strawberry in its young stages of development and you actually pick off all those developing achenes, all the dry fruit. So that's what they did in the middle. What they find is then that young developing strawberry stops developing. It just arrests. And that's what they're trying to show here. It just stopped. It's like, okay, I'm done. Now, if you do that same experiment, take a young strawberry, pick off all the dry fruit, and then spray it with auxin, it starts to develop more normally. So that's what they're showing on the right image. So that was one of the very simple experiment, but again, proving the point that auxin is necessary for proper fruit development. Now, since then, they've done all kinds of fancy experiments to further back this up, and it's still true today that yes, it does help promote fruit development and get it to maturity. So this is what I've been talking about, where if you cut off the apical meristem of the shoot, you can get side growth through the axillary bud. And so that phenomenon is called apical dominance. Apical dominance. So when you say that, what you're saying is that the shoot apex is intact, auxin is there, and what's really happening is auxin is flowing down from the apex down, and it is interacting with those axillary buds, and it's telling all those axillary buds, be quiet, don't do anything, stay dormant, let me grow, okay? And so the apex, the shoot tip will keep growing. If you cut off the apical meristem, then apical dominance stops, auxin stops flowing. The axillary buds say, I'm not being told to stop anymore, so then they start to grow and send off new shoots or leaves, depending on the plant. So that's what you're seeing here, where on the left, you see this shoot going up off the picture there, and then they cut it. So you can see right there, they amputated it. Now you see the axillary buds have sprouted new leaves right off it. All right, so this is one way that you can control the shape of your plant. If you want a bushier plant, you cut off the apical meristem, and it will release the apical dominance and allow for the axillary buds, the lateral buds, axillary buds and lateral buds. It's the same thing. Okay, so it is inhibiting auxin, actually inhibiting all the axillary buds. It's dominating, saying I get to grow. The shoot apex gets to grow. The axillary or lateral buds do not. But if you get rid of the auxin, then the axillary buds will sprout and start growing. Okay. All right, we'll stop there and continue on with plant growth on next Tuesday. Have fun in lab.